Hello everyone, welcome back to another video. My name is Saurabh Pawar. Today video topic is central dogma of life. The central dogma is the process in which the genetic information flows from DNA to RNA it to make a functional product protein. In molecular biology, central dogma illustrates the flow of genetic information from DNA to RNA to protein. It is defined a process in which the information in DNA is converted into functional product. It is suggested that the information present in a DNA is essential for to make all proteins, and RNA acts as a messenger that carries information through the ribosome. Let's talk about what is the central dogma. The central dogma illustrates the flow of genetic information in the cells and the DNA replication and the coding of the RNA through the transcription process, and further RNA codes for the proteins by translation. The concept of sequence of interaction can be understood through the framework. The most common includes biopolymers. The major category of biopolymers includes proteins, RNA, and the DNA. That are the further divided into general transfer, unknown transfer, and spatial transfer. The spatial transfer occurs in exceptional cases in the laboratory. General transfer occurs in almost all cells. It describes the regular flow information through the transcription and translation. Unknown transfers are said never to occur. The new DNA strands are formed with the one strand of the parent DNA and the other is newly synthesized. This process is called as semi-convergent DNA replication. Let's talk about the central dogma steps. The central dogma takes place in two different steps. First one is the transcription. Transcription is the process by which the information is transferred from one strand of DNA to RNA by the enzyme RNA polymerase. The DNA strand which undergoes this process consists of three parts, namely promoter, structural gene, and the terminator. The DNA strand that synthesizes the RNA called as template strand, and the other strand is called as coding strand. The DNA dependent RNA polymerase binds to the promoter and catalyzes the polymerization in the 3' to 5' direction. As it approaches the terminator sequence, it terminates and releases the newly synthesized RNA strands. The newly released RNA strands further undergoes post-transcriptional modification. The next step is translation. Translation is the process by which the RNA codes for specific protein. It is active process which requires energy. This energy provided by the changed tRNA molecule. Ribosomes initiate the translation process. The ribosomes consist of larger subunit and smaller subunit. The larger subunit in turn consists of tRNA molecule placed close enough so that the peptide bond can be formed at the expense of energy. The mRNA enters the smaller subunit, which is then held by tRNA molecule of the complementary codons present in the larger subunit. Thus, the two codons are held by two tRNA molecules placed close to each other, and the peptide bond is formed between them. As this process repeats, the long polypeptide chains of amino acid are synthesized. Genetic code. Genetic code contains the information of protein manufactured from RNA. These are basically 3 nucleotides and 4 nitrogenous bases which collectively form a triplet codons that codes for one amino acid. Therefore, the number of possible amino acid ranges to 4 by 4 by 4 is equal to 64 amino acids. There are 20 naturally existing amino acids. The genetic code degenerates. This was explained by the features of genetic code, according to which few amino acids are coded by more than one codon, thus the causing them degenerate. Each codon codes for only one specific amino acid, and codes are universal irrespective of the types of organism. Out of 64 codons, 3 are stop codons which stop the process of transcription and one of the codons in an initiator codons, that is AUG coding for methionine. Thank you.